There is always a risk of bioequivalence, there is a risk of stability, and there is a risk of manufacturing process for the generic formulations. So, the generic formulations are required to be safe and effective, comparable to the reference formulation. Bioequivalence is one of the requirement for the generics to demonstrate the similar safety and efficacy compared to the reference formulation. There is a risk involved in the bioequivalence study for the generic formulations, as well as there is a risk of bioequivalence study or bioequivalence outcome for the formulations made with some changes or variations. Also, whenever there are some spec changes, there are some moderate changes or major changes, the risk of bioequivalence study will be there. I welcome all of you to this video, and it is a very interesting video for all the professionals. Let's start with some basic information. Bioequivalence studies are the required studies to be demonstrated for the generic formulations to prove similarity to the reference product. The bioequivalence study mainly for the oral formulations will be discussed in this video, and bioequivalence study are those studies which demonstrate the similar rate of absorption and similar extent of absorption of the API from the generic formulation to the reference formulation. The risk assessment of the bioequivalence study helps to identify the factors that may have impact onto the study outcomes and ensuring regulatory compliance and the successful BE demonstration. Always, the generic formulations are manufactured, and you can say these are designed and developed in a very similar way or very comparable to the reference formulation. That may be the size shape, that may be the resolution, that may be the other quality parameters. Designing the generic formulation involves the studies and the parameters considerations to be in line with the reference formulation. Now we will see the risk for these parameters one by one. First is the formulation related risk. Formulation is made up of API or drug substance, excipients, and manufacturing process, and also the packaging process and the packaging material. First of all, the API should be similar to the innovator or to the reference formulation. API properties like BCS class, polymorphism, particle size and morphology, and some other parameters like degradation behavior, GI stability, and many others have the risk. If the formulation contains highly soluble API, it can lower the risk of bioequivalence failure. BCS class 1 and 3 have the lower risk compared to the BCS class 2 and 4. If the formulation contains BCS class 2 or 4 API drug substance, then it becomes a risky formulation because the poorly soluble APIs may have a great risk of bioequivalence failure. Another is the polymorphism. Polymorphism affects the stability, solubility, bioavailability, and manufacturability of the formulation. That's why different polymorphs may affect dissolution and it may affect the bioavailability. That's why the bioequivalence risk will be there. So always try to use the same polymorphic form as like the innovator. Particle size and morphology. Variations in the particle size and morphology can affect the dissolution rate and also may affect the permeability. That's why always do some reverse engineering. Try to find out the particle size of the innovator to be in line with the reference formulation so that you can reduce some risk. Then formulation related risk for the bioequivalence. Excipient and composition. Excipients influence solubility, permeability or metabolism. Sometimes some excipients are there which affect the rate of absorption, permeation and the bioavailability. Some excipients are critical for the formulation like surfactants are there, buffers are there, pH modifying ingredients are there. Some solubilizing property of the excipients may have great impact. And these critical excipients must be assessed for their role in the drug release and the drug absorption. Always try to have the similar composition in line to the innovator. Do some reverse engineering, find out some critical excipient ranges, and then you can reduce some risk for the bioequivalence. Some excipients may behave differently in the formulation. Some materials or some excipients have multifunctional role like pre-gelatinized starch is there, HPMC material is there, some binders are there. These are binders as well and these improve the solubility as well. Sometimes the polymers reduce the precipitation in vivo. That's why the composition becomes very important and it is a part of risk assessment. Always try to match the Q1 Q2, that is qualitative and quantitative similarity of the composition to the innovator. Also try to make the same size and shape of the tablets or capsules. Always try to match the physical parameters so that you can reduce the risk. Manufacturing process. So process variations may impact drug release and this in turn will impact the bioavailability and bioequivalence. There might be some processes like granulation, compression, coating, which can affect the formulation drug release. Scale up changes should not alter the dissolution profiles because as we know the formulations are developed in the research lab on a lab scale and then these are transferred to the commercial scale. So some scale changes are there which affect the dissolution. Study design risk. So not only the formulation and API and manufacturing process, many times because of the selection of wrong study design, the bioequivalence study may fail. Selection of subjects, healthy volunteers versus patient population. For example, some narrow therapeutic drugs are there. Consideration of fasted versus fed state. Dosing and administration. Food effect studies if food influences the absorption. Multiple dose studies versus single dose studies based on the drug pharmacokinetics. If you choose the wrong study design, then there are high chances that the bioequivalence will fail. Pharmacokinetic parameters like Cmax, AUC, Tmax variations due to formulation or physiological condition may be there. Risk of high intra-subject variability with the CV of more than 30%, requiring crossover design or the replicate study. So selecting the proper study design, selecting the subjects are very important parameters. There is always a risk of study design. Analytical and statistical risk. Bioanalytical methodology can be risky for some of the formulations. Sensitivity and accuracy of LCMS and MS methods. Stability of analyte in the biological samples may add the risk. Statistical analysis, how the outliers are handled, how the dropouts are handled in the study. Power of the study to detect differences within regulatory limits of 80 to 125% for the CI. Then regulatory and compliance risk. Some risk are related to this section also. Compliance with the US FDA, EMA, ICS, WHO guidelines, country specific requirements for B study approvals. For some time, it is observed that the bioequivalence is passed or it is meeting the requirements, but because of the compliance issues, the study needs to be performed once again or repeated. Then ethical and safety considerations are there. Adverse event monitoring and subject withdrawal criteria should be clearly defined. Sometimes inclusion exclusion criteria should be defined. Informed consent process and the ethical approvals are the risk. Risk mitigation strategies. So as we understood the risk factors, now we will see the risk mitigation. First of all, it is the characterization of RLD and deciding the QTPP, CQS, CMS, CPPs, control strategy of the formulation. Optimize the formulation, match the dissolution profiles, always go for discriminatory dissolution profile, always match the dissolution profile in multimedia, make the formulation similar in the Q1, Q2, and then you can say that the risk is reduced to some extent. Always consider what type of formulation is being developed, whether it is generic formulation with infringing strategy or it is with the non-infringing strategy. Always check what are the similar points and what are the different points between the generic and the reference formulation. Then conduct pilot bioequivalence studies to identify potential variability and optimize the study design. You can have good IV-IVC data. You can correlate this data with your dissolutions so that you can reduce the risk of failure of the pivotal B studies. Robust bioanalytical validation. So this is one of the most important thing to ensure assay accuracy, specificity and stability of the bioanalytical samples. Proper statistical planning. Use adaptive study designs for highly variable drugs. Now you can use the adaptive design. You can clearly define the design criteria and you can go for the adaptive study design so that in a one shot you can do some planning, you can have some adaptive designs and you can complete the bioequivalence studies. I have made a video on adaptive study design. You can go through that video and understand what are the adaptive study designs for bioequivalence. Regulatory consultation. So consultation should be done with the regulatory authorities. You can arrange pre-submission meetings to align the expectations so that there should not be any surprise at the last moment. Conclusion of the video. Now we'll see a systematic risk assessment in the bioequivalence studies ensures reliable, reproducible results, reducing regulatory and clinical failures. Addressing formulation study design, analytical and regulatory risk proactively enhance study success and smoothens the generic drug approval. Also, there is always a risk of biowaiver for the additional strength. So, whenever you are performing the bioequivalence studies or you are making the product for global regions, always consider the guidelines for biowaiver for the additional strengths. Sometimes for some of the market, some bioequivalence studies may be different. So there is a risk of region specific guidelines also. Always have a closer idea about these guidelines and the differences between the region specific requirements. I hope this video will help you to understand the risk assessment for bioequivalence studies. Always, whenever you are working onto the generic formulation, try to be in line with the reference product. Do all the risk assessments, do all the reverse engineering work, and then come up with the robust formulation. Thank you for watching the video, and if you have any comments, doubts or queries, you can mail me and you can comment also. See, I am also a learner for many of the topics. I am learning by making these videos. So don't consider me as a very well versed person in this field. I am a learner and I am learning by making these videos. I, I am very much thankful to the viewers for their comments and appreciating me to the views and comments. Thank you.